Hi, I'm Nigel Reuel. I'm an assistant professor of chemical and biological engineering here at Iowa State University, and it's a privilege to talk about some of the research we're doing in the area of diagnostics and other sensors that might be relevant to your work. So if interested, please be in contact with us. So the first thing I want to show is what are kind of the core ability of our group, and that's to look at passive sensors in closed systems. And this slide shows a smattering of the different publications and work areas. And I also give kind of a crude diagram here to show what a sensor is. You have an analyte, a thing you're trying to sense. It requires a selective domain that's then connected to some piece that can relay that information. We call that a transducer. Now, typically the transducer is wired directly to a reader, but in a lot of cases, that wiring is inconvenient. And so we think about systems where you want a wireless contact-free method, so that way you can sense something in an environment that doesn't allow for wiring. And some of those are agriculture, cell manufacturing, human health wearables. I wanted also to start with a slide here on a little conflict of interest in our what we do with technology transfer. So I'm a founder of 3 different companies: Scroot Laboratory Incorporated, which focuses on wireless process analytical technologies for biomanufacturing, located here in Ames, growing as it's at the seed stage. And then 2 companies that are just emerging right now: Frugi Biotechnology Incorporated, that focuses on low-cost diagnostics, a lot of what we're going to discuss today, and then Zymosense Incorporated, which looks at enzyme design and probes for sensing. So this slide I like because it shows kind of a diagram of the electromagnetic spectrum and areas where you could actually penetrate in and be able to extract information from a closed system. So obviously X-rays, we're all aware of how those work, but those high energy systems are often have disadvantages for the integrity of biology and materials. So we look at this near-infrared area, which we use nanoparticle probes, and then we also have these resonance sensors, which work in the shortwave radio frequency. And both of them, again, can be used in closed environments where aqueous tissue, where, again, it might not be convenient to have a wired connection. So I'll mostly talk today about our application of RF-based sensors for diagnostics, but I wanted to show a slide here of our use of near-infrared probes. So they emit in the near-infrared, and this little claymation movie here that I have playing shows that when you excite a nanotube with a light laser, that it fluoresces, and then when something selectively binds to it, it will cause a fluorescence shift, a change. And then when something non-selectively binds, it doesn't cause that shift. So we exploit the sensitivity of a nanotube to create a very small sensor. And again, we're using this for enzyme design, as well as looking at protein detection. But this next slide is really what I want to focus, or kind of the core technology of our diagnostics, and that is using shortwave radio frequency, where we have this coil that has a resonant frequency, and it's sensitive to the local dielectric. And so if that changes, and we can engineer the system such that it changes, we'll get a shift in resonant frequency, and we can measure that at a step-off distance. So here I want to show the problem that we're all very aware of with diagnostics right now, which is kind of threefold. So a picture of this line of cars. If you have had to wait to get a diagnostic check, especially for COVID-19, you're well aware of this bottleneck, that it takes a trained person to take your sample, and the next photo on the slide shows the reagents that are used. So you have a swab, you have a tube, you have reagents, and at the early stage, especially of COVID-19, there was a huge lack of these materials and of PPE that's needed to the personal protective equipment. And then the third bottleneck is all of those samples then have to be taken to a lab that has specialized equipment to slowly assess by a trained person if those samples have the virus or have the pathogen that you're trying to detect. So we thought about these and thought about what are ways that we could overcome this with our platform. So this is a drawing of this schematic, this dream that we had, that you would have a sensor, a card, where you could take it home and that you could put a sample on it from saliva and then have it cure, if you will, just incubate at your house and then placing it into an envelope and then bringing it back to a place of work or school where it could just be rapidly scanned. Now, you could have it such that you could read it at home, but there's an advantage to having a centralized reader, especially if it's fast and anonymous. So if you have this closed envelope and only you get a text message of the result, that anonymity is there, but also the ability for a school or a business or public health being able to track outbreak and correlate that to individuals is very important. And so we wanted to preserve that. So kind of stepping through the technology that we wanted to put into this is this RF coil, this resonance sensor, being able to have it have a coating that selectively degrades when the virus is present. So how do we do that? So the thought was that we could put in a toehold switch, which is a genetic element developed by a team in Harvard, where if a target piece of RNA is present, it will bind to it and cause it such that it turns on. And when it turns on, you can then express a protein. And they usually express something that changes color, again, something that you could read out at home. But the question we had is, could we express a protein that then has an activity that we can read out on this resonant card? If so, then again, that card could just go into an envelope. So here's a diagram of all the different steps that has to be done in order to achieve this dream. And I'm happy to say that we've done most of them to this point. We still have some on the sample collection and application part that we're still working on. But this idea of being able to take a virus, pull the RNA from it, amplify it, have it be sensed by a toehold regulator, make a protein, and then read it on a resonator is all possible. And so we're excited for this. So some of the background work that Sadaf has done is looking at a coated resonator, so something that has a coat of gelatin on it, putting on bacterial protease, and then seeing that we get a very nice signal shift. So in order to make that protein, we leverage something called cell-free protein synthesis, an area of work that we've been very excited about and then been able to get into from the side of practicality. How can you create a extract that creates protein with high reliability and with high efficiency and low cost? And so a cell-free reaction is essentially what the name says. You take a cell and we're getting rid of the cell by opening it up and just using the stuff that's inside the cell to make a protein. So that's called extract. You also need a DNA template, this gene template, and then you need supplements. So that cell brings over a lot of the components needed to run the reaction, but we have to add some energy and cofactors and things to keep that reaction running. So Jared, my student in this space, has done a great work looking at each of these three spots, and I'll just touch on them. So he's done a few papers looking at scalable extract. So how do you make a lot of this extract at a very low cost? How do you make an extract that can do disulfide bonds? And then also, how do you make a template that's minimalistic, so very small, has all the critical elements to make a reporter protein, but nothing extraneous? The work on the toehold switch has been done in collaboration with Alexander Green, who was the original inventor of it during his time as a postdoc. He and his very talented student, Kevin, were able to make sequences, design them such that there's this switch that if it sees different parts of, in this case, our target is SARS-CoV-2, that if parts of that RNA are present, they'll bind and then they'll create our reporter protein, which in this case is subtilisin, a very active protease used heavily in industry for detergents, so break up those protein stains. But in this case, it's creating subtilisin whenever that SARS-CoV-2 is present. And so if now that's made, The question is how can we can read that? And that's been the work of Adam. So Adam has created a scanner. So in our minds, we're thinking something like a fax machine type size where you put in your envelope and it would go through and read. He developed this scanner which reduces the positional noise. And he's done a lot of modeling work to determine the optimal size of this resonant coil as well as where to place that coating. Because when that switch is hit and it creates subdolysine and starts degrading, we want that to have the biggest signal as possible. And so he's done a lot of modeling and also experimental work to determine that. And these lower pictures show kind of the form factor. So it's a very small envelope. It could be made bigger if you want it to be. But it's, again, something that somebody could take home and administer on their own and then bring back and scan. So this slide 18 is kind of where our exciting latest results are, as we've shown that, that it works. So we can take this card, we apply a sample, it sits to incubate for a little while, and then when it dries out and you take it and put it in that envelope and scan it, so if you took it back the next morning to your place of work, it can selectively see the target as well as a control. So that's what this actin is. It's a protein that you're going to have in your saliva anyway, so that lets us see, okay, you did the test correctly. But then we also have another switch present to see if you have the virus, in this case SARS-CoV-2. And the exciting part of this technology is it's very modular. So you could think of that card being something that could be mass-produced and at a low cost and just kind of put on a shelf. And then the only thing that has to change is that toe-hold switch. So that part that recognizes some virus, once we know what virus target it is, it's pretty easy to come up with a new one and then embed that into your, as a add-on, as a layer to that card. This next slide also shows an idea that we have of other use cases where maybe you do want to detect it at home or on site. Let's say you're in the middle of somewhere where they don't have access to a VNA or a reader for it. We've also shown that you can have these lateral flow strips where you can just add your sample, it degrades, and then it causes this gelatin to move down the lane, and you get a very clear signal if it's present or not. And again, that could be useful to tell you, but in the broad scheme of things, we'd also want, if you have a centralized reader, you're able to track. So next steps for our pandemic test are again, we want to demonstrate this with lysed attenuated virus, what we're doing now. And we're looking at follow-on research funding to transfer this. So this is what Frugi Biotech is going to work on. And again, just as I have here written, we're seeking burning applications. So if you know of a human, animal, or plant pathogen that would benefit from a low-cost pen-side or bedside or field-side test, we'd be very excited and seeking to talk to you about it. I was told to end with a clear ask of kind of what our group needs and wants. And really it's in lots of different ways we can work together. So they ask us, if you find any of our technology interesting, let's find a way to do it. I kind of have a list of ways that we've worked with other corporate partners, kind of in layered increased involvement to decreased. Of course, there's corporate research. I've written grants where industry partners are co-PIs and we do research together. Hosting interns, the NSF pays for the students' time and they're able to be embedded in your company. And of course, technology transfer and working through the startups. If one of these technologies is mature enough that you're looking for direct application, I'm always happy to make those connections with the right people and discussing those opportunities. Must end with a slide here of everybody together. So this is our COVID pandemic picture. We're adequately spaced. I have a very good group of students and collaborators, and of course I need to acknowledge funding in this case that we derive both from federal sources, foundations, and corporations. So, and always looking for others who again can help us take our technology and apply it in some way that it would actually impact the world. So thank you.